Hi everyone. So this is welcome to CamBRAIN Journal Club. We're very excited to have Yizhou here today, presenting his research on Parp mutations and how they protect from mitochondrial toxicity in Alzheimer's. So, Yizhou, the floor is yours. Take it away.
Yep. Thank you very much for bringing me to talk to you. So I'm Izu. I'm a PhD student from Miguel Martin's lab at the University of Cambridge, and I'll talk to you a bit about PAR mutations and Alzheimer's disease. So first, I just wanted to say a bit about the background. Alzheimer's disease is very prevalent. Is the most common form of neurodegenerative diseases, and about 1 in 6 people above the age of 80 will eventually develop Alzheimer's disease. So how does the pathology work? Right now, I think the most prevalent consensus is that first you have the accumulation of this toxic misfolded protein called amyloid beta. And then this eventually is associated with neurodegeneration at a later stage of life. And finally, you have the manifestation, the effect of this neurodegeneration, which is cognitive impairment, which when your brain and your function cannot be normally performed. So my talk is, my research is mostly targeted at the accumulation of toxic amyloid beta. And basically, I'm trying to figure out how to prevent amyloid toxicity to lead to neurodegeneration. And to achieve this end, I would just want to say a few words about how this amyloid beta toxicity occurs in human. There's this amyloidogenic pathway that leads to the accumulation of toxic amyloid beta. And briefly, very briefly, there's a beta secretase and a gamma secretase that cleave this amyloid precursor protein. And this leads to the production of amyloid beta 42 amino acids. And then this accumulates and eventually forms plaques around the neurons, so extracellularly. And then this leads to diseased neurons and eventually neurodegeneration in human. So my work first uses flies as a model organism. And briefly, how I modeled this accumulation of toxic amyloid beta is by using this GAL4-US system. And the GAL4-US system is basically very simple. It's in simple words that this GAL4 protein is expressed under the control of a specific driver, such as LOV. And LOV expresses this GAL4 protein paneuronally, so in neurons mostly. And then this GAL4 protein will go on and activate this human form of amyloid beta gene. And then this leads to the accumulation of toxic amyloid beta outside of neurons. So this is the model that I'm working with. So using this model, I just wanted to analyze and explore the phenotype of this model. And one way that I personally very like is to do metabolomics. And metabolomics is the analysis of many metabolites, at least as many metabolites as possible in these flies, and compare them to healthy control flies. We detected about more than 400 metabolites in our samples, and then we used an in silico computational workflow to look at what processes in the brain in these flies are altered. And we found that nicotinamide metabolism is most influenced in in the flies expressing toxic amyloid beta. And then we looked at basically what metabolite in this process is the most affected. And the central metabolite is NAD, nicotinamide adenine dinucleotide, and it's decreased by more than twofolds. So it's half in the A beta expressing flies compared to control. And this metabolite is very, very important for mitochondria, for mitochondrial function. So then I hypothesized that perhaps there's some mitochondrial function dysfunction in flies overexpressing toxic amyloid beta. So then I looked further into that and literally took images of mitochondria in flies overexpressing toxic amyloid beta. And I saw using electron microscopy that flies overexpressing toxic amyloid beta have altered mitochondrial structure, mitochondrial ultrastructure. In fact, we can see that their cristae is degenerate, looks different than the control ones, where the cristae is very dense, whereas this cristae from flies overexpressing toxic amyloid beta seem to be fragmented. And indeed, after some quantification, we do see that flies, the diseased flies have a high proportion of fragmented mitochondria, with fragmented cristae. And then we used to follow up and to validate this observation, we used TMRM, which is a dye that fluoresces, and it's correlated with mitochondrial membrane potential. And membrane potential is very, very important for mitochondrial function. So it could be a readout of mitochondrial function. And we see indeed that flies overexpressing amyloid beta have decreased mitochondrial membrane potential, which suggests, again, that they have some dysfunction in their mitochondria. And then obviously amyloid beta, as I explained, could lead to neurodegeneration. So I also quantified that in flies. This is not really novel finding, but we just validate this, that it's also that neurodegeneration also occurs in flies. And how we looked at that is quite simple. In fact, there are rhabdomers, which are specialized neurons in the photoreceptor. So they are essentially eye neurons, in simple words, of the flies. And we see that in healthy controls, usually we see 7 neurons, 7 rhabdomers, but in flies overexpressing toxic amyloid beta, we see less. So, for example, in this case, we see 6 instead of 7. And after some quantification, obviously there's more degenerate rhabdomers in flies overexpressing toxic amyloid beta. And finally, in terms of the behavior and lifespan of these flies, we also see that using this infrared system, which records the activity of the flies, we see that flies overexpressing toxic amyloid beta are more inactive. So they sleep more, essentially. And if we quantify this curve, we can see that they sleep more, especially during the light phase. So they have what I would interpret as daytime sleepiness. And this is a feature that's also observed in patients with Alzheimer's disease. So I'm quite confident now that this fly model of Alzheimer's disease overexpressing A beta is a fairly good model of Alzheimer's disease in humans. And without any surprises, flies overexpressing toxic amyloid beta also have a shorter lifespan as a result. So my hypothesis is that Okay, perhaps that the accumulation of toxic amyloid beta somehow leads to a decreased level of nicotinamide adenine dinucleotide, which is very important for mitochondria. And I also see some mitochondrial dysfunction. And then comes some neuronal dysfunction and neurodegeneration, which leads to the flies not being able to stay awake for longer and then dying sooner than control flies. So then I tried to prevent this from happening. And one way to prevent this is by mutating this protein called Parp. And what Parp does is that Parp uses NAD to repair DNA in both flies and humans. So essentially, the hypothesis would be that by mutating Parp, then there will be more NAD for mitochondria. Therefore, mitochondria will be more able to deal with the accumulation of toxic amyloid beta. So then this is a bit of a busy slide, but we can just walk through it fairly quickly. The expression of toxic amyloid beta, but with the Parp mutation. So introducing this Parp mutation validates my hypothesis that it leads to a higher level of NAD. So by mutating Parp, essentially these flies that overexpress amyloid beta have a higher level of NAD. And this leads to, in fact, more higher level of TMRM, which is a readout of mitochondrial membrane potential. So it seems that by mutating Parp, flies that overexpress amyloid beta have healthier mitochondria. And then again, I quantified the rhabdomers, which is readout of neurodegeneration. And then I observed that Parp flies have less degenerate rhabdomers. So they have at least more, by this readout, more neurons. And then in terms of behavior, Parp also protects against this excessive daytime sleepiness that I observed in flies overexpressing A beta. So in this AD model of flies, this fly model of AD. So they have they have less sleep during the day. And then I also looked at lifespan, and you see that it does rescue a bit the lifespan as well. So the hypothesis here, the observation here is that introducing this Parp mutation seems to be protective against amyloid beta toxicity in the fly models. So then I looked at human because I'm modeling essentially the Alzheimer's disease in flies for humans. So in humans, I used data from the UK Biobank, which contains more than 500,000 people. So it's medical records from more than 500,000 people. And there's genomic information in many of these participants. So then I looked at all of SNP's single nucleotide polymorphisms, so all single nucleotide mutations in the Parp gene in humans. And I found that 3 Parp mutations, which are here, affect the risk of getting Alzheimer's disease. And how I looked at that is that I used first the whole cohort, which is basically comparing whether a person has Alzheimer's disease or not, and then whether there's a Parp mutation that affects this relationship. And then I looked at high-risk individuals. Basically, how I defined high risk is high genetic risk, because there are previous studies that showed that mutations in other genes also affect that the risk of getting Alzheimer's disease. So I looked especially in individuals with higher genetic risk for Alzheimer's disease. And interestingly, I found that is the same single nucleotide polymorphism, which is fairly good because this means that this relationship, the observation that this relationship is stronger, is more convincing in the high risk individuals. And in the low risk individuals, I found a different single nucleotide polymorphism. And this is a validation again that the diagnosis of Alzheimer's disease. So in Alzheimer's disease patients, there's a higher risk of sleepiness during the day. So this beta values is basically a sleepiness score that quantifies how sleepy a person is during. And I saw that another gene in Alzheimer's disease patients. So this is just looking at Alzheimer's disease patients and then controlling for various confounder variables. And I found another single nucleotide polymorphism that's associated with less sleepiness in Alzheimer's disease patients. So the hypothesis here is that perhaps future studies should look at these different polymorphism and their relationship to NAD levels, to Parp, and as well as Alzheimer's disease. So the conclusion here is that some Parp mutation does protect against Alzheimer's disease pathology in humans. But I also looked further aside from genetic mutations. And another aspect that I looked at is, why don't I just try to increase this level of NAD by feeding flies with NAD precursors, right? So by giving precursors of NAD, well, I was hoping to increase this NAD levels just by introducing like a pharmacological supplement to the food of these flies. So the supplement that I looked at is called nicotinamide, which is a precursor of NAD in flies and in humans. So indeed, I saw that feeding flies with NAM, nicotinamide, increases the level of NAD in flies, and then it increases the mitochondrial function, mitochondrial membrane potential in flies, again, overexpressing toxic amyloid beta, and then it protects against neurodegeneration in these flies. So nicotinamide, which is a form of vitamin B in humans, is neuroprotective, at least in flies. So then I also looked at human medical records, again from the UK Biobank. This is another fairly busy slide, but so on the left here, I used the whole cohort, data from the whole cohort, which is comparing healthy controls to people with Alzheimer's disease. And I saw that people who reported taking vitamin B seem to have a lower risk or odds of getting Alzheimer's disease, but this association is not significant. But then why I'm fairly confident with this association is that by subsetting people with a high genetic risk for Alzheimer's disease and looking at those people specifically, this association is stronger than looking at the whole cohort, which suggests that if you have a high burden, if you have a high risk of Alzheimer's disease and you take this drug, it's more effective if you have a normal risk of Alzheimer's disease and you take this drug. So that's a bit fairly encouraging towards that, towards the hypothesis that vitamin B is protective against Alzheimer's disease in humans. And I also looked at sleep, particularly in people with Alzheimer's disease, and then using again the whole cohort and the subset of people with higher genetic risk for Alzheimer's disease. So then after controlling for many confounding variables, I saw that vitamin B decreases the sleepiness score that I generated. And if we subset for people with high genetic risk for Alzheimer's disease, well, this strength of vitamin B to decrease sleepiness in humans is higher. So then the partial conclusion here is that perhaps vitamin B is protective in humans against Alzheimer's disease. At least it might have some effect. But obviously, this is an observational study. It's not a trial. It's not a clinical trial. So further study perhaps are required to validate this association with better controls. But this is, again, suggestive of a protective effect of vitamin B intake in humans against Alzheimer's disease. Okay, so I just wanted to thank other people in the lab and the co-authors of this work, particularly my supervisor, Miguel Martins, and other co-authors, Giorgio, Ivana, Sam, and other members of the lab who really helped me discuss and develop this work, particularly Rebecca, Marco, Nuno, Amrita, and Lydia. So really thank you for listening to me. And I was wondering if you have any questions. So yeah.
Thank you so much for the great talk. It was super interesting to hear about your research. So if anyone has any questions, please feel free to unmute and ask, or type it in chat and I'll read it out.
Yeah, so I think I'll just stop sharing.
Can I go?
Yeah, please go ahead.
So you said, Yizhou, that you are confident that these flies that overexpress A-beta are a good AD model. However, A-beta, only a very few number of patients actually have mutations on A-beta, right? The majority of the patients are sporadic. So can you comment on that?
Yeah, so actually, most of the patients in the UK biobank analysis do not have this mutation that I introduced in flies and develop our Alzheimer's disease by sporadic, and it's not a familiar form of Alzheimer's disease. And I also saw that in these patients, it's fairly there are associations that increased or at least taking vitamin B is protective in those people. So so the hypothesis would be that there could be a protective effect of vitamin B in humans as well, who might not have this mutation that leads to increased amyloid beta. But since increased amyloid beta is not only observed in people with mutations in the amyloid precursor protein, for example, that there could be an effect, that there could be increased amyloid beta in humans with the sporadic form of of Alzheimer's disease. So yeah.
Could it then be interesting, maybe give these drugs or do the Parp mutations in other neurodegeneration models, right? Or even tau I don't know if there is any fly tau model.
Yeah. So there are, in fact, many models of tau dysfunction in flies, and this is for sure some future work that we can pursue, that we should be pursuing further. And in other models of mitochondrial toxicity, like for example in Parkinson's disease, where mitochondrial toxicity and mitochondrial dysfunction have also been observed, mutations in this Parp gene and increasing NAD levels have also been found to be protective. So some preliminary evidence are indicate that um, mutating Parp or increasing NAD levels seem to be protective in flies with or in diseases with mitochondrial dysfunction. So I see a question in the chat. Shall I read it out?
Yeah, go ahead.
So the question is, how many point mutations were found in human which can connect to Alzheimer's disease? So in my study, I found 3. But there are obviously limitations to my workflow, which is basically it's dependent on how many mutations are found using this genomic approach, right? If you sequence genes from humans, in very simple words, then there are limited single nucleotide polymorphisms that you can find. Sometimes you can miss some fairly important mutations as well. So in my study and using my data, I found 3. But future studies with more comprehensive sequencing of this mutations in Parp could look at could be done essentially to improve this to improve our knowledge about the relationship between Parp and Alzheimer's disease. Oh, okay. Another question, which is, does sequencing panel, for example, 23andMe, for lots of DNA-based skill diseases exist for all of Alzheimer's mutation? Which company is it? I'm not sure if I understand your question. Oh, okay. So, oh, right. Yeah. So, so basically, I think your question is, are there genetic tools that you can use to check your risk for Alzheimer's disease? And the answer, the short answer is yes, but you might not want to. And the simple explanation is that, you know, throughout these years, many studies have shown that many single nucleotide polymorphisms, that many mutations are increase your risk of developing Alzheimer's disease. And these mutations have been found in many genes, not just genes related to amyloid beta. Right, so if you sequence your genes and technically and analyze it, right, using some in silico pipelines, then you can compare and you can predict your risk of developing Alzheimer's disease. And for example, in my models that I generated, you can use those models to predict your risk of developing Alzheimer's disease. But is this a good enough, you know, a good thing? Well, well, we're not sure because, well, I don't really believe that, because Alzheimer's disease has a huge environmental effect. And perhaps knowing that you have Alzheimer's, that you have high risk of Alzheimer's disease might be good, but for example, your education levels and your financial levels and your cognitive levels, if you have high, for example, cognitive levels, then perhaps you can, these factors are even more, I would say, or really important towards your risk of developing Alzheimer's disease. So yes, there's a genetic risk for developing Alzheimer's disease, and I've generated models for that in my work. But uh, uh, should you be worrying about that? I don't think so. What we should be doing, or what I think we should be doing, is improving our general health, which is uh, a huge predictor of getting Alzheimer's disease, like cardiovascular and cerebrovascular health, which are also very important predictor of or risk for developing Alzheimer's disease. And sleep, right? Yeah. Okay. There's another question by Cecilia. So, how long do you need to take vitamin B for it to be protective? So, this is a question that I haven't answered in my study. Um, so my simple answer to you is that I don't have these results, particularly in humans, because the answer to your question would involve a lot of money and a lot of time, and that will be a topic of a clinical trial to answer. So in simple words, I don't have the results to support any hypotheses or any suggestions to your question. Yeah. Okay. So can somebody prevent Alzheimer's disease if someone has high risk of Alzheimer's disease? Yeah. So as I tried to explain, Alzheimer's disease has, for example, a genetic component and an environmental component. So the genetic component you can't change, but the environmental component, that is something that you can change. And for example, people with high, I'm going to use a special term called high cognitive reserve, which is if you use your brain a lot, and if you, for example, use your brain on a daily basis, uh, if your work involves a lot of thinking, then the hypothesis will be that you are training your neurons and you're increasing this concept called cognitive reserve. And this is one of the concept. And another hypothesis is that Alzheimer's disease has a huge factor influencing Alzheimer's disease is your cerebrovascular health. So, for example, if you have a high waist to hip ratio, if you have a high body fat level, okay, that will increase your risk of developing Alzheimer's disease. And that risk, people have quantified it, is sometimes higher than your genetic risk. So unless you have this familial form of Alzheimer's disease, which is direct mutations in this protein called APP, the amyloid precursor protein, okay, and this is very, very rare. It's only 0.1, I think, percent of the cases were very, it's it's a very rare mutation. So unless you have that, then the rest of the risk of developing Alzheimer's disease, to my knowledge, would be mostly things that you can influence in addition to your genetic risk. So the short answer is yes. you can prevent Alzheimer's disease if you have a high genetic risk by taking care of your brain and your body's health. Okay. I think Niha, we've and
yeah, are there any other questions from anyone? Just if you want to message or unmute anyone else.
yeah. So if you have any other questions, I think you can email or message me directly. Or I think many people saw the tweet on Twitter, so you can directly message me on Twitter if you have any other questions. Okay.
Okay, in that case, thank you so much, Yizhou. I hope everyone enjoyed the talk. I'm sure they did. And see you next week in our next journal club. Thank you very much.
Yeah, thank you. Thank you very much for having me. Yeah, thank you, everyone.
See you.
Yeah.